sistema le avisará cuando comience la emisión. La emisión está comenzando. Todos los asistentes están en modo de solo escucha.
Hi.
Hello, can you hear me?
Yes.
I can.
That's wonderful. Yeah, so great that you organized everything and that we can really do it. So I hope you can see my presentations.
Yes, we can see it.
Yes, we can see it.
Very good. So yeah, then we are ready to go, right?
So, Carla, are you there?
Yes, hi.
Yes.
So can we make it like in this order that I will, hi.
Hi.
I can see now you, Abraham, but I cannot see the Connie's webcam.
I don't have my webcam on, so maybe
Okay.
so I can do it, but I was afraid that the line is not strong enough. So I think the priority would be with the slides, and then if I finish, I can put it on. So this is what I experienced the last days or weeks, so that I should not do both at the same time in my internet.
Okay, I will try to switch on my webcam, Andre. Ciao. Hello. So, Carla, can we organize in a way that I will start with a short introduction, then we pass to Abraham, he will present then Connie, and then Connie's lecture. And then I think all the participants can ask questions online.
Yes, they can. We can not program to pass one slide to another slide, but I can give control to the people in question. And you can also, because you're an organizer. So, for example, if you stick with the...
if I start now, can you share my... so how can I share my... just wait, maybe like no?
Yes, right now. I just made you presentator.
Yeah, okay, because this is the I see. I can make presenter Abraham Cornelius Zaballa. Okay, so
yes.
If I know, how can I get on? Okay, just wait. I'm trying to.
You have the control now.
Okay. So I would like to do some of the things. Okay. So now you can see, for example, this this one.
No, we can see anything, Gabri.
How can I share then my screen? I mean the PPT presentation. I will make myself as a presenter.
I
am not
I
here.
already made you. Wait a second. I will do it again.
Okay.
Now you have to. Yes. Now we can see.
I'm closing a few things because I cannot see the pop-up of my presentation at the moment. So maybe this one. So that may be like this. So you can see it now, the presentation only.
The presentation and the screen. So the video.
you can see the presentation and the
and my screen and you in the video and the webcam.
okay. Okay, I think it's okay like this one. Is that right?
Yes, for me it seems yes.
So Connie, is it okay for you? So after that one, I will make you as a presenter. I mean Abraham as a presenter, and then Connie. I think it should work, shouldn't it, Carla?
Yes, it should work. Do you want me to do the naming of the presentation or the presentator? I can do it myself if you prefer.
I will do that, I think, because when I will make this short introduction about the seminar series, I will say that Abraham is going to present Connie, and Connie is going to give the lecture.
Okay. Okay.
Yeah. Maybe there is going to be a little bit of little bit of mess when we select the presentator, but shouldn't be so much, no?
No, it's not hard. You just have to click with the right button at the top of the name and just click on make presentator, and that's it.
Okay. So how many we are online now? Let's say
21 plus the 4 of us.
Mm-hmm. I think we wait another 5 minutes and then we can start. What do you think? Connie, how are you? Hello? Connie?
I'm sorry, I just turned off the mic. So I'm
oh,
fine.
You're scared.
Yes.
Okay, I would like to make a quick trial to switch between me and you, maybe. So now I will make you presenter like now.
Okay.
And then you can switch on, I mean, your presentation, I think.
Yeah, so you should see it. Now I tried. Let me see. So you see it?
Okay, perfect. Yes.
Perfect. Wonderful. That works.
Okay. Sometimes we have technical problems, and it's mainly because I am not so much good or quick at. But actually, I think this time it will work. So we are all at home. It's a nicely relaxing environment. Abraham, how are you?
Yes, I'm fine.
Yeah, so we have like a lockdown in university as well. So we do not work much at the university at the moment, as everyone.
Yes, unfortunately, yes. So I was really very happy that you accepted this invitation and make it online, the presentation, Connie, because with the last 3 ones, I had a little bit of difficulties because obviously it just this emergency with the COVID just happened, and people were shocked, scared, or whatever. So it was very difficult to organize at the beginning. I mean, this webinar. So 3 of these seminars, which were foreseen in the original plan, has been postponed at the end of the series. And this is why I would like to show also to the people who join us online today these changes. So this is why I would prefer to start with a short introduction, and then Abraham will present you, because he knows you very well. And then yeah, obviously I would be very happy if people could ask also questions using the the board. I mean,
that sounds really
written
good.
questions. I mean, if I don't know whether you are going to read these questions, or I can read it for you. If you had we will see as you wish. We will see
Yes, how can I see them? Is it appropriate? It pops up and then I see it and I can answer it, right?
That's a question section on the panel. And if you click on the arrow, then you can you will see that.
Gabby, I think we can only see them because both of us are organizers.
Okay.
So only you and
I
me.
don't see. Then you probably need to read them.
Okay,
Yes,
then I will read it and then you will answer it. I will try to read it clearly and slowly. So,
okay. Otherwise, I'm gonna ask you again.
Okay, yeah. Okay, so all the attendees are online now. Carla, what do you think? Or we still have some time? When who decides?
There were like 50 people who register, and right now there are 30. So maybe wait 5 minutes to see if more people connect, and then just start. What do you think?
And when I start, you do, or when you, so for example, now we are online, or they are listening us or not. Okay, that's good. That's good.
They are
so
listening
they are
to us. Yes.
okay. No, it was not clear. I thought it would be very, yeah, only only 4 of us.
No.
Okay. Okay. So maybe then I can speak to all. So we will wait, as Carla suggested, another few minutes just to have some more attendees join online, and then we will try to start right after that. Okay, so we are 40 people around, so I think we can start. So good morning and welcome to the seminar series on extracellular vesicles. I am Gabriela Fox-Falvi, the organizer of this seminar series, and here is our program today. So after this short welcome and introduction, Abram, a PhD student of Professor Cornelia Kex, will introduce his supervisor, and then we will have the seminar of Professor Keck, which is followed by a discussion section. I just would like to point out that if you would like to ask Professor Keck any questions, you can type them in the questions tab within your control panel. So I just would like to show the aims of this seminar series to people, to those attendees who are the first time joining us. So the main aim was to bring together researchers who are interested in extracellular vesicle research. So the seminars, which included the series of seminars, which includes 10 seminars, were organized on every Wednesday at 12 p.m. And these last 2 seminars, which remained, will be only available online. So the main aim of the seminar series is to promote collaboration in a Campania region of Italy, but also collaboration, international collaboration. We are interested in all types of sciences, so some of the lectures mainly point out some basic topic in extracellular vesicles, where others are mainly focused on applied aspects of extracellular vesicle research. We are interested in all type of vesicles. So in this seminar series, we are talking about small vesicles, extracellular vesicles, large vesicles, even plant-derived vesicles, which are not only extracellularly expressed. We talked about mammalian vesicles, also ancient type of cellular communication in hydrovesicles, also blood, urine. So we are really talking about a lot of different type of vesicles and trying to show the diversity of the vesicle field. We would like to supply an ambient space for discussion, so all your questions are really welcome. And I would like to point out that this seminar series is sponsored by the Italian Society of Extracellular Vesicles, called the VITA, and also by the VES4US European project. So everyone who is interested to join this seminar can use a link, which this link is available on the VES4US and the EVITA web pages. Well, we are also quite active on Twitter. So these are the two tweets what we placed for the today's events. So you can follow us in the Twitter, VES4US, Evita Society, and on. This is our program, and here you can see actually the 10 seminars which were planned from 5th of February till 8th of April. So you can see that until 4th of March, everything went okay. So we had regular seminars organized in vivo at IBBR CNR in Naples. And we transmitted all these seminars also online on the GoToWebinar platform. Then you can see these seminars, unfortunately, in red, which has been postponed. And then reason you can, I guess you know, it was the COVID-19 emergency. So due to this emergency, we really had to postpone this seminar at the end. When we will be able to transmit this one, you will know it from our websites and from our emails, what we will send regularly to all those people who are registered. Actually here we are today with this presentation of Professor Cornelia Keck on plant crystals. She's from the Philipps University of Marburg in Germany, but I would like to ask his PhD student, Abram, to give a short presentation of Cornelia's.
Okay, thank you, Gabri, for giving me the floor. So I'm happy today to go back to the seminar series. I was hoping it would be in Napoli, but unfortunately, in any way, it's good to have it again, even online. I'm happier actually today to present my supervisor, Dr. Cornelia Keck. She is a pharmacist and she has finished her pharmacy studies at Free University of Berlin. And then she has done her she has done a lot of movements actually. So she has done a postgraduate study at Otago University in New Zealand. Then in 2006, she has done her PhD studies in pharmaceutical sciences at Free University of Berlin. Then she continued at the same university with her postdoc studies in 2011, and she works as well at Pharmazol in Berlin as a manager and then as a head of the research and development department. She works as a professor at different universities in Germany and outside Germany, so I will mention only some of them. So she worked as a professor at University of Vienna in Germany. Then she was a visiting professor at University of Putra in Malaysia, and then at the University of Applied Sciences of Kaiserslautern in Germany. Since 2016, she is a professor of pharmaceutics and biopharmaceutics at Philips University in Marburg here, and she has done a lot of researches actually, and she has a long experience with researches, more than 18 years, I think, and her research field is about pharmaceutics and cosmetics and nutraceuticals nanotechnology. She focused on her lab on formulation of polysoluble actives using different nanocarriers like nanolipids, drug nanocrystals, and many others. And even she works as well on the tablet made from paper. I will not talk about the publications because they are really a lot, but I will mention that she has 5 patents, I think, and yeah, so a lot of publications. And one of the most interesting topics for me, at least, and I think for many, is the plant nanocrystals, or what we call planticrystals, which we will enjoy today to listen at. Thank you. And the floor is yours, Connie.
Thank you so much. This was really a nice introduction. Thank you very much. And also thank you so much for organizing this, even though we have that very special situation. So I try to share my screen. Is everyone seeing it?
Yes.
Okay, that's wonderful. Thank you. So when I got the introduction, I was really pleased to say, yes, I really want to come to Neapel, we say in German. And then unfortunately, I cannot be there, and I would really enjoy being there. So maybe we can postpone it to another time. And so today I only want to give a little introduction about the research we are doing, because I really look forward to have cooperations with people that really have a different field of expertise, because I think this is really the great thing in science, that we can share knowledge and combine knowledge to something very novel, something very special. So this is why I choose to have a little mixture of what we are doing, and then of course I focus on the plant crystals. But before doing this, I think it's not running. Let me check. Okay. So probably not everyone knows where Marburg really is. So Marburg, can you see my mouse? Maybe.
Yes.
So that's wonderful. So then this is Europe, and then you see, so it's in the middle of Germany. And if you enlarge it a little bit, then you might have heard something about Frankfurt. So this is the largest airport, and we have a lot of money, financial stuff in Frankfurt. So very close to Frankfurt, this is where we have Marburg. Marburg is a very, very old town, so it's really useful coming there because you can enjoy it. It's not, like Italy, but as long it is a beautiful city. So it's around a valley, which is called Lahn. So it's a small river, but it makes the city a little bit more relaxed in comparison to other cities. We have a small population of about 70,000 people. And people would say it's not really a city, it's the university. So many cities have university, but Marburg says we are university. And so if you look, yes, and if you look at this, it's we have a very old university, has been founded about 500 years ago by a landgrave, and his name was Philip, and this is why we are named Philip's University. It's the oldest operating Protestant university in the world, and we have about 25 students and about 10,000 employees. And then if you look at how many people are working, so it's 50% of the whole population, and this is why it makes sense to say Marburg is the university. So, and then of course we have many different studies we can do. So I belong to the School of Pharmacy, and we have a very large pharmacy school. So we employ about 280 students each year, which really is a lot of fun. So I like teaching a lot, and so Marburg is perfect for this one. And then I work in the Department of Pharmaceutics and Biopharmaceutics. We are 2 professors. So one is Professor Barkowski, and then my lab. And what we are doing is we try to formulate nanocarriers with the aim to improve the bioavailability of polysoluble actives. And I would like to take the opportunity to explain a little bit about this one. So we say we do smart nanocarriers, and one platform of the nanocarriers are the plant crystals. So we do drug delivery, and this means we are interested in really transporting a pharmaceutical active to the correct place, so that means the site of action. We want to deliver it in the real correct dose at the correct time. So everything else, if you do not have it at the right place, it's the wrong dose or the wrong time, is not a good drug delivery, because that means the therapy will not work. So for transportation, it's like in normal traffic, you need to have a carrier. And of course, it needs to be a suitable carrier who really transports your active to what I said before, the place in the correct dose and time. So if you would go to a pharmacy, then of course, you know, there are a lot of dosage bombs which are available already. So we have tablets, we have capsules and so forth. But sometimes you have the problem that all the classical approaches are not successful in drug delivery. And then of course, in this case, you need smarter carriers to achieve your goals. And so there is one major problem in pharmaceutics today, and this is if you have a new chemical compound coming from synthesis, so very often is this poorly soluble. And this means you cannot use it in the body, because only if this, you know, the active dissolves in water, then you have the molecule, and only molecules can be taken up by the body. So this is a major problem. On the slide here, you say it's you see it's 70%, but actually can be up to 90%. So this is where we have a problem, and this is what we try to address. We want to improve solubility of such poorly soluble compounds. And we have different platforms. We have the so-called smart lipids, the smart crystals, smart pearls, and smart films, and the plant crystals. And I will shortly introduce some of them. And before starting, so you all know, because all the vesicles you're using are in nano as well. Nano in Latin is DARF. It comes from Latin and from nanos. And in English is DARF. In German, it means Zwerg. Of course, this is not scientific. So in science, we say a nano is a billion of a whole. So you see on the side, this is our Earth, and the Earth has a size of about 12,000 kilometers in diameter. So a nano Earth, a billionth of the whole Earth, is as large as a peanut M&M. So of course, this is a totally different dimension. And this is, I think, bringing a good example into the place, saying like, well, if you have nano, it really differs in properties in comparison to normal sized material. And so this is why we do nano. And here is the most prominent example, and many people use it. So what you see is a gecko, and you probably know that the gecko can walk on walls, it can walk head over heel, and this is because of his nanostructures on the feet, what you see here. So they are below 1 micron, and this means, and we can go to this side here, he distributes his whole weight to many different attaching points. So this means, in comparison to one larger thing, that the weight which has to be carried by one attaching point is of course less in comparison to larger material. And this is common for all nanoparticles. The smaller the size, the higher is the adhesiveness. So this is if you want to have something which really sticks to something in a much better way. So then you can use nanocarriers. And in regard to pharmaceutics, it means you have an increased adhesion, and by having an increased adhesion, you have a longer retention time. And if you think of an active needs to have time to penetrate from A to B, so of course, the longer it stays, the better will be the penetration into, for example, the bloodstream. And this means you have more active in your body, which we say is an increased bioavailability. So adhesiveness is valid for all nanoparticles. But then, of course, you see it in the upper picture. So we have many types of DARFs, and that, of course, means we also have many different types of nanoparticles. And so each particle, of course, has very specific properties. And for the particles we are using, I would like to introduce a little bit the specific properties of them. And I would like to start with the smart lipids, which we say is the third generation of the lipid nanoparticles. And the lipid nanoparticles have been invented in the 1990s, independently on by two different persons. So one is Maria Rosa Gasco from Turin, so Italy, one person, and Professor Rainer Müller from Berlin. This is the group where I did my PhD. So a lipid nanoparticle, and this should be visualized here, is composed of a lipid matrix, which is solid, at room temperature and body temperature. And in that solid lipid matrix, you can incorporate actives, typically lipophilic actives. And then, of course, you need to stabilize your particles, so it needs to be surrounded by a surfactant. So as I said, over time, some more types of lipid nanoparticles have been developed, and we are using the smart lipid concept. I will not go into detail. I just want to say in general, all the lipid nanoparticles are valid to encapsulate lipophilic actives. You can increase the chemical stability because you prevent them to come in contact with some oxygen, water, or whatever. And you have that increased adhesion, and this is why you can also increase the bioavailability. Typically in an oral way. But what we found is very interesting. If you use your lipid nanoparticles and you put them on skin, then they form a film. And because they are nano, remember that peanut MLN is the nano earth, so they are really tiny. You cannot see them by your eye. And so it's an invisible patch. But the invisible patch, and you can see it a little bit here, so it on top of the skin, and this is why the skin, is sort of sweating, so you lose water all day long. But if you have that little film on it, then of course the evaporation is not as strong as before. So this is why you have some so-called occlusion, which means you have an increased skin hydration. And by having more skin hydrated, the skin swells a little bit. So that gives you less wrinkles. It's interesting for cosmetics. But in pharmaceutics, it's very interesting because you have an increased dermal penetration, because you have more space between the skin cells. So all in all, the film formation also makes a skin barrier protection, so it helps a little bit to repair an impaired stratum corneum. And this is what we are doing. So here are some slides. And I don't see this slide, but I tried to put all the photographs of the PhD students really currently working on this project. So this is Sabrina, and she works on lipid nanoparticles, also the 3rd generation. And so you see this is an oil, you have an active in the oil, and this is skin biopsies, and then the slices of it. So this is the dye, and you see there is some penetration into the stratum corneum, but nothing below. Oops. And then you have a nano emulsion. So this is just oil droplets, a nano size. It's a little bit penetration, but you see a much more pronounced penetration of the active if you really use the lipid nanoparticles. So this is the principle. And then we have something novel. So VES4US is working on this project. Many people, and you have seen the lipid nanoparticles have been invented about 30 years ago. So this is not novel anymore. And so people say lipophilic actives are really good to be incorporated. And what we are now doing is we incorporate hydrophilic actives. And you see that's also working. So we have 2 lipid nano formulation, and we have a lipophilic dye is penetrating, but also the hydrophilic one is penetrating. And what is interesting is not only promoting the transportation of the active into the skin, is also promoting to put active into the hair follicles, where they remain a little longer. So you have a depot, and then from that depot, the active can be delivered. Either it goes transfollicular in this route, or it is pushed out again and then can be used for penetration via the stratum corneum. And how do we do it? So we say we do smart lipids with a hydro pocket, which means in a classical approach, we form a water in oil, a water emulsion, but because the solid solidifies if it's getting below its melting point, so we have a solid lipid matrix, and in the solid lipid matrix we have little water droplets, and in this water droplets we can have the hydrophilic actives. And so the smart thing about this one is we can now combine hydrophilic and lipophilic actives. So the lipophilic one would be in the lipid matrix, and the hydrophilic one would be in the hydrophilic little droplets. So you can have a co-delivery of both hydrophilic and lipophilic actives in only one carrier, and they are separated, and that's quite interesting, especially if you look at vitamin C and vitamin E. They need to be co-delivered, but they destroy each other if they are in one formulation at the same time and if they get in contact. So this is very current research, and we found this is very interesting. So the lipid nanoparticles are on the market for more than 30 years, or no, more than 20 years. And no one really had looked at the interplay between the lipid matrix composition and the biopharmaceutical performance. And we now found, and actually it was sort of an accident too, that there is a very strong influence of the lipid matrix composition on the biopharmaceutical performance. So here is one example. We have more examples in regard to biopharmaceutical performance, but to guide you a little bit through the slide. So this is a nanoemulsion, which means the lipid matrix is composed of 100% liquid lipid. If we go to the other side, then we have solid lipid nanoparticles. So this is the first generation of lipid nanoparticles being composed of 100% solid lipid. And then, of course, you can have mixtures. So, and this is what you have here, a whole range coming from high amount of liquid lipid going to low amount of liquid lipid. And what we then did is we incorporated a fluorescent dye into the particles, and then we checked again for penetration efficacy into the dermis. And you really see the strong influence on the lipid matrix composition. So what we want to have in the future now is to really understand this interplay, and then having tailor-made particles where we can really say this is a particle which repairs the stratum corneum, this is a particle which actually promotes penetration, and by maybe mixing them we can have both. So the second formulation principle we have are the smart crystals, and this is something very simple because it's the second generation of the nanocrystals. A nanocrystal is, you see it here, it's a crystalline material composed of 100% active, and the only difference to normal material is it's a nano size. And you can get the nanocrystals very easily by either bead milling, high pressure homogenization, or by combination methods. The inventors are here up, so it's Gary Liversidge and Rainer Müller. And so also here you see it's not really a novel formulation principle, but you can really do a lot with them. And the beauty about this technology is it is so simple. You take the material and then you put it in a homogenizer or in a mill, and you get out nano-milled material with a nano size. So in regard to the nanocrystals, it's very simple. By having this, you increase the solubility. And this is because if you look at this, so this is a large crystal, then you have a small size. And if you would look at the surface, you would see that the curvature is really increasing, which means the smaller the size, the higher is the curvature. And if you go for the Kelvin equation, then it is clear that the vapor pressure depends on the curvature of your particle. So the smaller the particle, the higher is the vapor pressure because of the increased curvature. So there is, more pressure to evaporate. And we can transfer this principle to the nanocrystals when we say, okay, we have an increased curvature, and it's not the vapor pressure, it's also the dissolution pressure. So the tendency to go into solution, into a solvent. And this is working. And then you see on this side the Noyce-Whitney equation, and I really don't want to go in detail, but what it is saying, so if you have a higher surface area, so this is the surface area, and by having it in a smaller size, you have a larger surface area. So then, of course, you increase the dissolution velocity. You increase also the concentration gradient, because if you have a higher solubility, then of course you have an increased penetration gradient, and this is what you can see on the next slide. So low solubility, concentration gradient low. In contrast, nanocrystals high solubility, this means a high concentration gradient. And most of the actives in pharmaceutics are taken up in the body by passive diffusion, which means an active goes from A to B until it reaches an equilibrium. And of course, if you have a high concentration gradient, you have a higher passive diffusion, which means an increase in bioavailability. So this is why nanocrystals are very good for poorly soluble actives. So I want to give you one example, and now we are coming a little bit more to the plant crystal and the biological material. So we are very much interested not only in chemical compounds, but we are very much interested in secondary plant substances. So there are flavonoids and many other things, and they all possess poor solubility. And then, of course, the idea is by transferring the normal material into nanocrystals, you can increase the bioactivity. So here is one example. It's routine. You see it here in the, I don't see it, but I think it's here. There should be a pansy. And so if you see a pansy very close to the ocean, then it's quite purple. But if you go to Alps or to mountains and you will look for pansies, then you will see that they become more yellowish in the flower. And the reason for that is that the plant is creating rutin as a protectant from UV light. So it's a UV protectant, and Beiersdorf, which is a large cosmetic industry, already found that effect many, many years ago. But then they also found that the rutin is polysoluble. And what they did is what you see here. So they make a chemical synthesis, and then they produced rutin glycosid. So sugar makes a polysoluble compound more water soluble. And they could show that if you use that compound in 5% weight into a formulation, you can increase the UV protection for skin. So what it is here is the so-called sun protection factor has been measured in a human study. So you go to UV light, it's a simulator, and you can irradiate the skin with the precise dose of UV light. And then you measure the dose which is required to get a little sunburn, which is called minimal erythema dose. So that little sunburn and the dose needed to get this one. So you see this is untreated skin is set to 100, and by using that rutin glycosid, then you increase by 30%. So then, even though I don't see it, but should be another graph, and it should be by about 160%, and it's routine in a 0.01 concentration. So that means you have 500-fold less routine in comparison to that old formulation, but you have a double effect. So that means 1,000-fold better in comparison to an old molecule, and this is what the nanocrystals can do. So we are much interested in really transforming this knowledge into products which can really help patients. And of course, a pharmaceutical product would be wonderful, but of course it's much, much easier to show that the technology is really doing something if you go for cosmetic products. This is why we have done some cosmetics products as well. And so here you see skin biopsies. This is a punch and with normal skin, and you see that little dots. And the little dots, they are Langerhans cells. So very sensitive cells. And if you radiate the skin with UV light, then you see it looks different because it gives an immune reaction, which means that the Langerhans cells, they go down to the skin, so they look less in this biopsy, and then they transform a little bit because of that immune response. So in comparison, you see here the biopsy which has been taken with the same UV dose as this one, but before the UV dose, they were treated with the nanocrystals. And there's almost no difference to the untreated skin. So this is what nanocrystals can do if you use a UV protectant. Of course, they can do more. And so many years, people were just doing the nanocrystals, and we now started to look at the final formulation. So of course, you can imagine a nanocrystal formulated is like a watery suspension, like milk, and you have to formulate it to bring it on skin. And so the point I would like to make is really, really important in which, cream base or gel you incorporate your formulation. So, and this is true for all nanocarriers and everything. So it's not only that your system is nice, you have to look at the environment to really get the whole sketch. So in this case, we suggest that the nanocrystals should go in a mixed system which contains oil and some water and some emulsifier. So the nanocrystals, of course, are also interesting for other things. So if you look for the secondary plant substances, they are highly antioxidative because of the poor solubility. You have to increase solubility to get more antioxidants done. And so you can, of course, use antioxidants for the treatment of oxidative stress-related diseases. So this is Alzheimer's disease, many cardiovascular diseases, but it's also Parkinson. And so that has been done in a cell culture model for Parkinson disease. So we have a control. This is the neuronal network of cells, neuronal cells. And then you see if you add a protein, which is synuclein, and this is typically created in Alzheimer patients, then you see, so this is the numbers here, that it breaks down the network. So the cells are dying, and this is what happens if you develop Alzheimer disease. And if you add antioxidants, then you see that's becoming better. And you see that micro is giving some action, but that the nanoroutine, and you see it here, so it's getting more closer to a healthy control. This is how we can use the nanocrystals. And before I continue, I would like to show you something about another technology that we think is very interesting, because you will later on understand that this might be the technology to make the plant crystals even more attractive. So many people work with porous material to increase the solubility of polysoluble active, and so they use porous silica and microparticles and everything. And we were wondering if we can use something else which contains pores. And we wondered if paper can be used to load active into the pores, and if it is staying amorphous. And because if you have something in amorphous state, you increase the solubility. And I mean, it was really not logic that that would work, but for any reason it worked. So we really have a patent application on that. We can load polysoluble actives in normal paper, and we can prevent the amorphous state, thus leading to increased solubility of a polysoluble active. So we see here again routine, and this is nanocrystals, this is bulk material. And so you see it's not as high as the nanocrystals, but this is because we have a little filter effect, because not everything is coming out of the paper again. But you see it's increasing solubility as well. That's working. As you have probably seen already, we really do a lot on skin experiments, but you see it is much more effective than the nanocrystals. This is the nanocrystals. This is curcumin. It's a very poorly soluble compound, not penetrating in bulk material. Nanocrystals do it a little bit better, but you see a very intense penetration into stratum corneum and a very nice transdermal penetration of the curcumin. So this is what the smart films can do on skin. But the question is, would you eat paper? Probably not, because it's probably not so tasty. And so this is why we had now done a very interesting study, and we could show, well, no problem. If you cut the paper in little pieces, put it in a tablet press, you receive wonderful tablets, and they are conformed to all the requirements according to the European Pharmacopoeia. So that is hardness, resistance to crashing, and mass uniformity, friability, and also things. So then we were interested in really transforming this technology into a large-scale production, and that's simple. So you can transform the paper into pellets. Then you have something which is flowable. Then you put the pellets into the tablet press. This is a so-called filling shoe. And from the filling shoe, it rinses into the matrix, and then you have that automatic production. And at the end, you get tablets made from paper. And they really look like normal tablets. You don't have any differences to normal ones. But actually, pelletization is not so easy. And this is why we now found a very simple way. So we take the paper, we make the smart film, we mill it, and then of course it's powder, and then you can compress the powder into tablets. And you see there is no influence. The smart film and the tablet, they have the same solubility. So also this is working. And we think this is really a very nice approach to transform something polysoluble in something which is nicely soluble. But in addition, you might have seen this is a blank paper, and you add a liquid and you let it dry. So we think everything which is liquid can be dried on a paper, and then you have an easy method to dry something without any lyophilization or spray drying, which are all expensive and energy consuming. So, but now I have talked so many times about secondary plant substances, and of course the smart carriers help to improve their efficacy. But then, we were thinking of, plants produce such interesting compounds, and you don't have any problems by having that in the plants. So maybe nature is really smarter than our nanocarriers, and maybe we can do something being inspired by nature. And this is where we came up with the plant crystal technology, saying like, we do something where we combine nature and our, knowledge and experience from the nanosciences. And so we wanted to have whole plants or plant material, and of course this is herbal medicines. And as every medicine, so you should heal, improve, or prevent diseases. And so we have school medicine, we have alternative ones, and we have the phytopharmaceuticals. We have different ones. So we have one where we know already they are really working, so in a scientific way, and we know which ingredients are responsible for that. And this is the compounds we are working at the moment with. So if you have curcumin extracted from a plant, then of course, you know, the curcumin is the ingredient being responsible for all of things. But we have many phytopharmaceuticals where we do not know why is it working, or we know maybe it is working, but there is no scientific proof. And still, so many people use it and say, this is really helping me for this and this disease. So plant material is really interesting in many, many ways. And there's a very long history as well, and you probably know better than me. So it's not only in Europe. So in Germany, we have Hildegard von Bingen. It's more than 500 years that she started to write down all the knowledge she had about herbal medicines. Then of course we have Ayurveda, we have traditional Chinese medicine, and there's so many different treasures, and it really needs to be preserved and it needs to be used as well. So I think there's a lot of demand. We can do things better, not only with school medicine. Of course, many people use phytopharmaceuticals, so it's also attractive to make them. And it's not old-fashioned anymore. So if you look at phytopharmaceuticals which are sold today, it's not only a tea, it's actually every dosage form we have available can be used to formulate phytopharmaceuticals. So we have tablets, we have pellets, we have capsules, everything is possible. So how do we produce phytochemicals today? So tea is something old-fashioned, and some people still use it, but actually what we do is we do extracts from plants. And extracts from plants is really large scale. And so the principle, even though I do not see it, is very often the same. So you have a percolation, which means you have two different mechanisms that occur during the extraction. So one is a direct washing out, and the other one is because you have cells that are intact. So you need a diffusion. So extraction takes time because you need to swell your plant material to let all the extracts, the compounds coming out of the plant material. So, and this is very important, of course, if you have your plant material, you have a lot of waste as well. So I have visited some factories that produce herbal medicines, and it's really a shame how many tons of plant materials after the extraction are discarded because there is no use anymore. So, and this is what we wanted to do. We wanted to have plant material, and we wanted to produce something which is more effective and which is environmentally friendly, and which does not produce so much waste. And we think the plant crystal technology is a nice solution. It's very simple. So you take your plant, typically dried plant material, you grind it, and then you apply some forces like bead milling or high pressure homogenization mixers and so forth, and you get suspensions, watery suspensions, and depending on how much forces you apply, you can really scale it down to the nano dimension. So you get really nano plant particles, and the name plant crystals is just coming from the nano crystal approach because we use exactly the same techniques. And so plant crystals are parts of plants or plants in micro and nano dimension. And so what we are interested in is really to use plants, mainly medical plants, and we want to use them from different geographical regions. And we really want to learn about history, about all the knowledge in medical therapy from plants, because many things are going to vanish. And I think this is really something important. And by increasing efficacy of these traditional plants, so that could be really help to have little plants, but make most out of it. So what can we produce? So we can take the whole plant, we can take peels and also pomace or mark. So this is, you know, the leftovers, as for example, if you do wine. So the peels, typically they are discarded. But if you go for, you know, the French paradoxon or many other things, we know that especially in the peels, there's the highest amount of all these interesting compounds. So it's a waste, but for us, it's really turning waste into value. But probably everyone who have met Abraham before, so he says or tells everyone, this is what we want to do. We want to turn waste into value. So I explained that before. So we have that washing out and we have that extraction phase in traditional extraction. And if we nanonize, so one should know that the size of a cell is approximately 10 micrometer. So if we go in a nano size, this means we can be really sure that each cell is destroyed. This means we have no washing out anymore. And so this is wrong. And we have only, no, we have the washing out directly because we have destroyed all the cells and no extraction, diffusion is necessary anymore. So, and the principle of extraction is very simple. So it's high pressure homogenization. You can imagine high pressure homogenization by thinking of a syringe. And then you fill in the liquid into the syringe and then you squeeze it out. But you try to avoid squeezing out quickly by putting up your thumb on that syringe. And if you really press very closely to that, you know, to really bring out the fluid, and of course you increase pressure and some liquid will go through the gap between the syringe and your thumb. But then if you look closely at it, then the distance the water or your liquid is flowing is much faster. So this is the principle of high pressure homogenization. You have a high pressure, you accelerate the liquid, and by having this one, there is a so-called Bernoulli principle. And I think this is too complex. If someone is really interested in the technology, I'm happy to explain it later. In any way, you really destroy all the particles. And so it's called cavitation. And so what we think is we really destroy all plant material, but of course, and this is, you are the expert and I'm, you know, a beginner. So there are exosomes in plants and they are derived from different origins. But if you have such a vesicle like here, and they're, you know, they are not in a constant shape, so they are sort of an associate, and they associate depending on the environment in different sizes and different layers. And I go back one slide, 2 slides. So here we know that if you have a liposome, which is an artificial vesicle, we know that we can change size and also structure of liposomes by high pressure homogenization. And so we are pretty evident that if you use high pressure homogenization plant materials so that we can modify the vesicles which are in the plant material, and we would be so happy to learn more about this one, because if you have the vesicles, then we know that all the liposomes and the vesicles which people are using in the pharmaceutical field are used to encapsulate actives, and mainly polysoluble actives. And we do it to increase the bioavailability and efficacy. So we are exactly back to the topic we are interested in. And so, of course, we have no experience and also no availability for any instruments needed to investigate it further. But I think if we can measure and we can adjust the exosomes from the plant material, we can further improve the efficacy of our plant crystals. So we have done, even though this is not yet done, and Abraham started in Italy, and hopefully we can continue very soon. So we already did quite a lot of work. So we homogenized many different plant materials, and Abraham is one of the most active ones really doing this. So here are some examples what we already homogenized, and here are some examples of results. So this is coffee waste. So if you cook your coffee, then of course you discard the leftover. But you look at, here is the antioxidant capacity, and it's expressed as the so-called IC50, which means the concentration needed to scavenge 50% of a free radical. Means the smaller the IC50, the higher is the antioxidant capacity, because you need only little to scavenge the free radicals. So this is the nano of the the coffee, so nanosuspension of coffee waste, and you see it's very close to ascorbic acid, and ascorbic acid, as you know, is a very strong antioxidant. So then we have here banana peel, and this is actually our first study because no one really knows or needs banana peel. Banana peel is really a waste and annoys many people, especially if you slip on it. But don't discard it. This is bulk, this is micro, this is nano, this is ascorbic acid. So yes, banana peel can be helpful to fight antioxidants. And so this is now getting a little bit more serious and more into traditional medicine. So this is sumac, and Abraham brought it from his country. So sumac is a very special spice. And again, you see by nanoionization, you nicely increase the antioxidant capacity, and it's similar to ascorbic acid. But the thing is that, of course, there is much more than only antioxidant capacity. Plants can really have more medicinal uses like antimicrobial, antifungal activity. And this is what we wanted to show in different experiments as well. So we have done different plants. Here are two examples, is the Maltesian mushroom and the mistletoe. And here are the results from the Maltesian mushroom. So we tested antimicrobial activity by measuring the viability of the bacteria. And so what you see, this is a plant extract, so traditional way to produce herbal medicines. And this is the nanoparticles. And especially at the lower concentrations, you see that the nanoparticles are more effective than the traditional plant extract, and also they are longer lasting. So means we have particles produced, we have no waste, we have no organic solvent, because the extracts, they really they need a lot of organic solvents, otherwise you cannot wash out all the ingredients, and still you have a higher activity. And this is very interesting because you can really do something. So we did some further studies to understand what we are doing. So we wanted to look at, is there really an improved extraction? So we have RIEM, and RIEM established some tests and measuring different antioxidative values. So, and here we did it with some special roses coming, I think, from Saudi Arabia, and they are typically used for perfumes. In any way, it's just an example showing there are differences, and it's very interesting to look at them. So Abraham produced the particles, and then we had bulk material, we had the microcrystals, we had the plant crystals, and we measured the amount of polyphenols in our system, and we could nicely show the smaller the size, the higher is the amount of polyphenols you extract from your system. So that was our first test. Then we did the same material, but we tested the flavonoids. And what you see, the same trend, but the trend is much more pronounced. And then we did another test. So we tested the total amount of carotenoids which are released during the homogenization, and the effect is even further pronounced. And this nicely shows the potential of the plant crystals. Because if you go from polyphenols going to flavonoids and then to carotenoids, there is like the carotenoids are more hydrophobic than the flavonoids and the polyphenols. So that means you have no problems doing extraction by extracting hydrophilic compounds. But if you destroy really all plant material, then you can also extract the lipophilic actives, and this without any organic solvents. So then, of course, we are interested in the biopharmaceutical efficacy, and we have this dermal model, and this is why we tested this again. So stinging needle has a lot of chlorophyll, and Dani Knot, so you see his slide on the other side, had an idea chlorophyll should be fluorescent. And that really worked. So what we could do is we follow the chlorophyll once we apply it on skin. So this is a traditional extract, a water extract. It's tea, some penetration of the chlorophyll, probably little extraction of the chlorophyll, and then bulk, macro, micro, submicron, and nano. And you can see there is really a better penetration, and this is because nano sticks better to the skin, and of course it has a higher release of the chlorophyll. So this is the summary. Plant crystals are composed of plants and parts of plants, milled to a nano dimension. It's a smart formulation principle because you can increase the biological activity of plant material. It is environmentally friendly because you need no organic solvents, you have less waste. And of course, if you can make more out of your plant, you also need less plant material. And especially in herbal plants, this is very often a problem to get enough material. So that can help as well. It's quite cost effective because the methods we are using are simple and long known. It's a universal approach because you can probably do it with every plant material you can find. And so we think environmentally friendly alternative to classical plant extracts. And we really hope that we can use it. And what we learned, especially from the stinging needle, if you have plant material and water dispersed, so that starts smelling quite quickly. And this is why we think that by combining the plant crystals with the paper, you have a very efficient method to dry your plant material and still the efficacy remains. But this is what we are currently trying to investigate. So I would like to point out that we think the applications are universal as well. So not only in the pharmaceutical field, but also for food supplement in the cosmetic field, and also in the agricultural, because if you have a compound which is maybe, you know, active as an herbicide, you can accelerate the efficacy. That would be a nice way to do as well. So thank you very much for your attention.
Thank you. Thank you, Connie, for this very interesting lecture that I think besides other thing, what you showed us, you showed also a very interesting and alternative way of the producing of nanovesicles from plant materials. So thank you really very much for this this talk. It is now open for questions, and I would like just have a look at what questions arrived. Let's say there is one question that if you can show or tell again the difference between the nano emulsion and the SLN.
Yeah, I think it's best if I show it. Yeah. So a nano emulsion means you have an emulsion is two liquids are dispersed in each other. And so if you have an oil in water emulsion, then you have oil droplets and they are surrounded by water. And a nano emulsion is an emulsion and only the size is below 1 micrometer. So typical in the size between 150 to 300 nanometers. So, and then the only difference to an SLN, so SLN stands for solid lipid nanoparticle, and it's very simple. So you produce an emulsion. The only difference is that the lipid you are using to produce that emulsion is solid at room temperature and body temperature. So you can produce your emulsion by heating your solid lipid. It becomes liquid. Then you produce at hot temperature a nano emulsion. And then you just let it cool down, and due to cooling, you know, you go below the melting point, and then of course your lipid solidifies. And then you have your solid lipid nanoparticle. And the same with all the other, we call them NLC for nanostructured lipid carriers. And this is should be NLC as well. So, and we just now started to mix liquid lipid and solid lipid, giving us these different structures. So does that answer your question?
Yes, I think so.
Good.
So just okay, thank you very much, Connie. So and another question from Mauro. Can you tune the dimension of your plant crystals by applied pressure, by changing the pressure, let's say?
That's a very good question. I would say not yet. So at the moment, no, we cannot, because we are still in the process to understand what are the major parameters influencing particle size. And it's not trivial. You know, for the nanocrystals, we meanwhile have that approach to do statistical approaches, so design of experiments to really tailor-made particle sizes. And so we are far away from that for the plant crystals, because if you look at plant material, then, or maybe from the other side, so if you look for high pressure homogenization, homogenization or any diminution process. So then, of course, we have to look at the properties of the material. So we talk about brittleness and elasticity, and fibers are different than, you know, shorter material, and plants are different to normal material because they contain too many different material sorts. So some, it's like little brittle, most of them are very elastic. And so I would say if we have a sort of standard plant material later on, we say, okay, then we can try to understand for this specific material how to modify and maybe tailor-made the particle size. But for general, you know, let's say we have roots, we have flowers, we have leaves, that might be approach later on to say it in this way. But a general approach at the moment, no, not yet.
Okay. Another question, do you check the actual composition or the identity of such small plant crystals?
In which regard? So I mean, we check that the plant we use is really the plant we want to use. That, I mean, this is something very important, especially if you get something very, you know, very rare stuff. So this is why, especially for the mushroom and everything, we asked experts to really make sure this is the plant. And now I got the question. No, yes, we really do a lot of efforts to investigate what is the influence of the homogenization or milling on the chemical composition of the plant material, because of course due to the milling you can destroy a lot of plants, and this is why we started to have so many different assays to measure not only antioxidant capacity but also flavonoid content and carotenoid content and so forth, because oxidation is a really issue, because if you diminute the size, then, as I said, for the nanocrystals, you increase the surface. And by having a larger surface, then probably the particles and also the compounds in the particles are more prone to oxidation. And so this is, of course, what we need to investigate, and we try our very best. But of course, there is much, much more to do in this regard.
Thank you. So another question from Rita Carotta. How do you control the increase of concentration of some toxic materials, like that could be present in the plant? So how can you control the concentration or activity increase?
Yes. This is it's a very nice question, and so the question for the question is, what is toxic? So if I have a medical plant, we want to increase the medical efficacy, and of course then you have toxic compounds. But as we know from Paracelsus, the dose is really giving you the effect, and so yes, we have to be aware what are the ingredients in each plant, and are we looking for maybe antioxidant capacity, and we also extract something different, maybe an antimicrobial activity? The only thing what is important to note is each active which is in the plant will be increased in regard to extraction if we use the plant crystal technology. And this means at the moment we don't have any, you know, specific method where we can say, like, well, we only want to extract lipophilic ones. We just destroy the plant material, as simple as that, and everything which is inside is coming out. And if, you know, there is something toxic or something we do not want to have in the plant, then we need to delete it later on by extraction from another method. But yes, of course, we increase toxicity if the plant is toxic.
Okay. Thank you. One more question, some more questions. One is that, Connie, please, can you suggest whether every plant materials can be processed as nanocrystals, or there are some specific characteristics required from Somas and Gupta? This is, I think,
yeah. Okay, so it's a good question, and I tried to answer that a little bit already. So the efficacy of milling has many parameters which are affected by that. So one is it's affected by the property of the material, and the other thing, it's affected by the milling method you're using. And so this is really complex, and so many people try to do it by trial and error. Some people try to do it by understanding. A mixture of both is the best way to achieve good results. So plant material is very often, depending on the part of plant you are using, very, very flexible. That means it's like a chewing gum. And then try to nanonize a chewing gum. So this is sort of hopeless. And so what you can try to do is to make the chewing gum and the plant material more brittle. And of course, this can be done by really freezing the material, for example, or by dissolving it in something, but this is for nanocrystals. No, in this case, you can do it by minimizing the temperature to make material more brittle. Or you use, and this is what we are doing, a combination of different techniques. So of course, you need to pre-mill your material. And so many people only use the pre-milling technology to decrease the main size to below 10 microns, because only if you have particles being below 10 microns, you can put it in a homogenizer. But what many people do not know is that the pre-milling has an effect on the crystallinity of your material. And this is also true for the plant material. So you increase the, I'm suffering from the English word, the order of your structure. It's imperfection. I wanted to say imperfection. So the imperfections are increasing. So you destroy a little bit your material, and by having something, so it's like, you know, you have a porcelain, like a plate at home, and it gets a little kink. It's still intact, but then for the next one, it will break very easily. And this is why it's quite important to use pre-milling techniques to do a little bit cracking of your plant material, and then typically you get smaller sizes and, more importantly, more homogeneous sizes.
Thank you, Connie. A question from Veronica. Could plant crystals be used to design a method for conservation of EVs? She's saying that the existing method for freezing is biased for some purposes, as thawing damages the EVs.
Can you repeat the question?
The question is that whether you think that the plant crystals can be applied or used to design a new method for the conservation of the EVs.
In which regard, to remain them as the particle size, or to store them more?
Because EVs are prone to they easily damaged or broken by during the centrifugation, or for when you are exposed externally. I don't know. So instead of freezing, if you think that plant crystals can be used, that's I don't know. Maybe Veronica can type some more details on this one.
Yes, maybe I start with what I understand what would be her interest, and then she can use the time to write more questions.
Yes,
well, I think yes and no. So by putting, I mean, I would just say, like, where we take some exosomes from somewhere else, and then we put them to the plants, and then we homogenize them. So yes, there could be the possibility, but we have to check. So it's just, you know, gut feeling now, that the EVs go to plant material, or at least adsorb to them, and then by homogenization they remodulate, giving you know, a different structure which might be more stable, but on the other hand can also be less stable. But of course there will be a change, and in which direction, to a better or worse one, I have no idea. But that could be really interesting to use nature, because it's not only that we have the plant material, because we have so many different compounds which are released and then, you know, still being in that mixture. So I think nature is really smarter than we are, and I think we should give it a go to test it, and I have a good feeling. So this is one thing. And then if that was not really the intention, and in regard to breaking and maybe also agglomeration, I think bringing normal formulations not in the plant crystal, but maybe in the smart film technology could be another option to do. So we take that paper, we have different sorts of paper, and then we just soak it with your formulation, and then we just look what happens. And especially we look what happens if we redisperse it, because that's also very interesting to see. It's not only that we preserve it for 1 minute, but, you know, typically you're interested what is coming out again.
Veronica say many thanks, so I think you answered perfectly her questions. That's, so let's see if some else. I think this was the last question. So if not, no more question, I would like to close this section. I would like to say many thank you, Connie, for this presentation and also answering all these questions. I also would like to say many thanks to Carla from Zabala for her very kind technical assistance. And of course, I would like to say many thanks to all the online participants. And yeah, I am pleased that you visited us, and see you next week. Bye-bye.
Bye. Thank you very much.
Thank you, Connie, indeed.
Bye.
Bye.